So let's do glucose plasma levels. So here's our fasting glucose levels. Here's our mealtime. We saw this in the first couple of weeks of our GI physiology. This is the uptake from the lumen, digestion. We got to the top, our maximal glucose concentration in our circulation. This is when insulin is having its main effects, binding to skeletal muscle, translocation to GLUT4, glucose uptake. This is also when we are in a high energy state. We're oxidizing glucose as much as we can to recharge our energy state because we're just coming out of a fasting state. And then we came to this curve. And so this is declining glucose levels in the blood. This is primarily due to GLUT4 activity on skeletal muscle. Adipose tissue also has GLUT4. Now this is the storage phase. So by the time we get to this level, which is approximately 90 to 120 minutes, our energy charge is high. So once our energy charge is high, then ATP can inhibit what glycolytic enzyme?
PFK1.
PFK1. And if we inhibit PFK1, where's the glucose going to go? So I do these kind of reviews periodically just to kind of reinforce some major principles. Glycogen synthesis, yeah. Glycogen synthesis. Now, under these conditions, is ATP completely inhibiting PFK1? Is this turning it off?
No.
No. So what else is happening? Fatty acid synthesis. What else is happening?
Pentose shots.
Pentose shunt.
Yeah, pentose shot.
The pentose shunt. We'll call it pentose shunt. So now, what this simply is the storage phase. So that's our number 3. Our ATP levels are high. Now we're shifting our glucose into storage, glycogen, glycogenesis, fatty acid synthesis, and the pentose shunt. What controls the pentose shunt? Nucleotides. Nucleotides controls the pentose shunt. You're correct. Where does it control?
PRP.
Yeah, PRPP. So that's the ribose, way down at the end. But what controls the entrance into the pentose shunt? Yeah, glucose 6-phosphate dehydrogenase. And that enzyme deficiency leads to you can't eat fava beans, yes. So if you ever see a question with fava beans in it,
6PD.
exactly, right? It's a deficiency in the enzyme. So what is the problem that is going to manifest in this deficiency? Increased reactive oxygen species. Is that going to be evident in all cells? Yes, because the pentose shunt occurs in all cells. And that means you're going to have an elevated oxidative damage in all cells. Now clinically, where is it going to show up?
Anemia.
Yeah. Hemolytic anemia. Is it going to show up in general anemia? What's anemia caused by?
Decreased hemoglobin.
Decreased hemoglobin, which could be we'll look at this next week, a porphyria, decrease in synthesis of one of the enzymes, or What's the most common cause of iron deficiency? Yeah, we'll look at that next week. So that's anemia, that's iron deficient anemia or porphyria. This is hemolytic anemia because these reactive oxygen species are damaging that red blood cell membrane. So G6P dehydrogenase deficiency affects all cells, but clinically it's going to show up in the RBCs. This is like that, when I do this, it's like when we were back in grade school. Pop quiz, pull out a clean piece of paper, put your name in the upper right hand. Remember that? We all hated that, right? So I'm just helping you relive your terror here. Okay, so now where are we today? We are right here at the bottom of this curve going out. Now what we've reached is our fasting state again. So at our fasting state, our blood glucose levels are now back to that 90 to 110 mg per deciliter concentration. And what is happening to insulin secretion? Down. So it's going down because, why would it go down?
Glucose is gone.
Glucose is gone. Mechanism?
Glycolysis.
Glycolysis was reduced in what kind of cells? Beta cells. So ATP levels are going down. What's happening to the potassium channel? No more inhibition, so it's opening up, so you're repolarizing the membrane so the calcium channel does what? Closes, and the vesicles for insulin secretion are no longer fusing with the membrane. So insulin secretion's going down, that's the short answer. What's that doing to the alpha cells in the islets?
Glucagon.
Is it encouraging glucagon secretion?
Yes.
And how? Does it bind, does it have an insulin receptor that says signal and cause glucagon secretion? You weren't expecting a pop quiz after a long weekend, huh?
No, it's a glucagon.
So you're relieving the block. Remember, insulin blocks the secretion of glucagon from the alpha cells. And by taking away the insulin, you are now releasing that block, and now glucagon secretion can occur. So glucagon secretion is a normal constitutive event. The only thing that's controlling it is the levels of insulin. So at this point now, we're starting to get an increase in glucagon secretion. So now what we're going to do is shift our metabolism 180 degrees. Because with insulin in the fed state, we were in an anabolic state. Now we have to be in a catabolic state. And again, this is the signal telling the body that we are now officially in a closed system. We have no other fuel availability, so we have to live with what we have inside. Okay? So, major shift in metabolism. So the first event that has to happen, primarily because of brain and red blood cell activity. So remember, the brain has GLUT3, red blood cells have GLUT1. So these glucose transporters will work under any concentration of glucose. But if our glucose concentration continues to go down, because remember, these transporters are working under all glucose concentrations, so they potentially could take just about all glucose out of the circulation. That means we will go into a coma when we get down to that 25 to 30 milligram per deciliter glucose concentration. So the primary event that we need to do as soon as we get into the fasting state is we need to control the amount of glucose in our circulation. And that is going to be done first by glycogenolysis. So we saw glycogenesis in the fed state. Now we're going to reverse our metabolism and do glycogenolysis. So glycogenolysis is the same. The pattern of metabolism, the events, the reactions are the same in all cells. So the first step is to take our glycogen, and we are going to do a phosphorylasis reaction. And a phosphorylasis is different than a phosphorylation. A phosphorylation is adding a phosphate to a protein using ATP. We're going to use an inorganic phosphate, and we are going to convert the glucose that's in the polymer chain into glucose 1-phosphate. Now, the enzyme that does this reaction is the most important enzyme you need to know in glycogenolysis. And this is called glycogen phosphorylase. And the reason this is important is because this is where the major portions of glycogen storage diseases occur, which we'll see in the next hour. So once we have glucose 1-phosphate, are we going to make galactose? Why? because it would be stupid.
Glucose 6.
But this is a trick. I would never do this to you on an exam because it's really a trick question. Because I would say on an exam, well, you need to make sugar, right? In a fasting state. So you have an opportunity to take glucose 1-phosphate and go backwards, right, in galactose synthesis. And galactose is a sugar, right? Energy source? What's the enzyme that prevents this from going backwards? The one that you have to know for the galactosemia. The name that everybody laughed at? Yeah, uridylyl transferase, right. So that enzyme is using UDP glucose and UDP galactose conversion, and that's what prevents this from going to galactose. So now, in the fasting state, you can see how important that enzyme is, that it's not reversible. You do not want to send this to galactose. You want to do this. And this is the phosphoglucomutase enzyme. This is the same enzyme that makes glycogen. It's just reversing the path. Now, once we have glucose 6-phosphate, this is where the liver and skeletal muscle part ways. So, in order to keep our glucose levels up in our fasting state, the glycogen stores in liver are going to be responsible for keeping our plasma glucose levels up. So the glycogen in liver can't use its own glycogen. It's going to convert the glycogen to glucose 6-phosphate, and then it is going to turn this into glucose using the enzyme glucose 6-phosphatase, simply taking that phosphate off that 6-carbon of glucose, turning it into glucose, and then now, because the glycogen is being reversed and you're making so much glucose in the liver, you can actually reverse the activity of GLUT2. And so this is how the liver puts the glucose into the circulation immediately at the fed state. Now skeletal muscle, yes?
Is this glycogen from the liver?
This is liver glycogen, yes.
So it is using its own glycogen to turn it into glucose, to put it into the blood, but it's not breaking it down.
Correct, exactly. So when we're in the fasting state, the purpose of glycogen in the liver is solely to supply glucose to the circulation. Okay? And then this glucose that goes out in the circulation is going to support the brain, red blood cells, kidney, any organs that are highly metabolic that continually need glucose oxidation for energy. Now, the muscle is a bit selfish. So this process of glycogen breakdown in muscle occurs exactly the same way until we get to glucose 6-phosphate. Muscle doesn't care that your glucose levels are dropping in the circulation. Simply because where is GLUT4 in skeletal muscle in a fasting state? It's retracted from the cell surface, yeah. So without the glucose transporter on the cell surface of muscle could care less if there's glucose or not. So it's going to take all of its glycogen and convert this into fructose 6-phosphate, and then on down glycolysis to make ATP. So that's the glycogenolysis, the couple of reactions involved. At glucose 6-phosphate, we have a branch in the road. Liver will turn this into glucose. Muscle will use that for energy production. Now here's a trick question that you'll see on step 1. I've heard the students tell me about this in the past, is that glucose 6-phosphatase, that's the key element here. So one of the questions will be, why skeletal muscle cannot produce glucose and send it out into the blood?
They don't have that enzyme.
Yeah, skeletal muscle does not have that enzyme.
And that's the reason why complete gluconeogenesis can only happen in the liver, right?
Exactly. This is the enzyme we're going to see tomorrow, I believe, in gluconeogenesis. The phosphatase activity is the final step in gluconeogenesis. Okay? So now, that's simple. Now let's look at regulation, because regulation is everything, and it differs between different tissues. So in skeletal muscle needs its glucose in a fasting state. But skeletal muscle also knows that it needs it distributed in a controlled manner. Yes, question?
For G6P, the fructose 6P, what's the enzyme?
Oh, that's phosphoglucoisomerase.
Okay.
Yeah.
Is that important?
Nope.
Okay.
I don't think there's any diseases anywhere near that. So the only mutases and isomerases, there's a lot of those in all the pathways. I don't recall any diseases being there because those are not points of regulation. So only the regulated enzymes are key. So I said glycogen phosphorylase would be very important to you. Oh yeah. We're going to see in our case today that that's a very important enzyme. And we'll see it here in regulation. So when skeletal muscle is in a fasting state, it wants to release some of its glycogen stores as glucose for energy. But what if you're just kind of sitting here? You're breathing, you're moving around a little bit, maybe you're taking some notes. What's going to happen to your ATP and AMP levels?
ATP will go down, AMP will go up.
Yeah, ATP is going to go down, AMP is going to go up. So this is a signal. It's an energy charge in the cell, and the energy charge in the skeletal muscle is going to tell your glycogen phosphorylase it's going to tell your enzyme to activate. So once you activate glycogen phosphorylase, now you're going to start removing some glucose. And then that's going to move, because you're in a fasting state, it's going to move into glycolysis. And the sole purpose of this is to reverse your AMP to keep your ATP levels up. Now what happens if you get up and start moving? You start walking around. Well, you've increased your neural activity. And neural innervation of skeletal muscle does what to calcium? I was expecting a resounding answer from everybody. Remember that neuroblock thing? Calcium, what does neural innervation skeletal muscle do?
Influx of calcium.
Causes calcium go up? What's calcium for? Contraction, good. So we're going to have another signal here with calcium. What calcium is going to do is it's going to activate phosphorylase kinase. And phosphorylase kinase is going to have two activities. It's going to add a phosphate to glycogen phosphorylase, and this will become even more active. So it's already somewhat activated by AMP binding to it, an allosteric activator. But now we're going to phosphorylate it with this kinase due to the elevated calcium signal. And we're going to turn the enzyme up even more. And so now what we have is we have an energy charge and neural innervation that is controlling metabolism because now we're moving around more. And that's saying we need more glucose to keep our ATP levels up. The other thing that phosphorylase kinase is going to do is it's going to phosphorylate glycogen synthase. The reason we're going to do that is because this reaction here, phosphate adding to glycogen to release glucose 1-phosphate, if glycogen synthase enzyme is active, what can we do with glucose 1-phosphate? Resynthesize glycogen. Is that what your skeletal muscle wants to do? No. So this one signal of this phosphorylase kinase is actually activating the phosphorylase and deactivating the synthase. Now, what happens on Friday, September 15th at 8:15 in the morning that you see the metabolism exam? You die. I was thinking of the fight or flight reaction. The first thing you want to do is beat the hell out of me for putting together such an ugly looking exam, but I'm not here. So your fight response isn't there. I'm seeing some people going, yeah. So the flight response comes in. So what you're going to do is you're going to jump up in the air and you're going to haul out of this room screaming. Yep. I'd rather work at a kiosk in the mall, right?
Which happens.
So that's... You know what's really weird and unprofessional that I almost did to my 423 class? When I handed out the exam one year, I thought about stapling a application to like McDonald's. Isn't that awful?
Have you when you do that?
Yeah. The tar and feathering would be awful. It would be terrible. So you're terrified. And so what's the hormone that jumps up? Epi, adrenaline, epinephrine. So epinephrine levels start to jump. We're going to hear a lot more about that in the endo block. So what epinephrine is going to do is it's going to bind to its receptor, which is a heterotrimeric G protein receptor. This is going to activate G-alpha-S. This will activate adenylate cyclase. This will increase cyclic AMP, our usual cascade. And this is going to activate protein kinase A. And guess what protein kinase A is going to do?
Phosphorylate kinase.
Yeah, it's going to phosphorylate the phosphorylase kinase, which is going to phosphorylate the glycogen phosphorylase. Yeah, blah. That's a tongue twister. And what's it going to do to glycogenolysis?
Decrease.
Increase. So what you're doing here is you've got a 3-stage. It's like turn the volume up on a stereo. You get a little bit of AMP, you want a little bit of glucose release from your stores. You get some neural activity now, you want a little bit more because there's more activity. And now with epinephrine, pedal to the metal. Let's just activate this whole system as far as we can go. Glucagon has no effect on skeletal muscle at all, ever. Why would it?
No receptors.
No receptor. The glucagon receptor is not present on skeletal muscle. The epinephrine receptor is, because you need to make sure that your skeletal muscle is prepared energy-wise for a fight or flight event. Besides, skeletal muscle, again, doesn't care about the plasma glucose levels. Glucagon is the signal. The name of glucagon came from glucose. And so the liver really cares about blood glucose level. So what the liver is going to do is it's going to activate glucagon signaling through the glucagon receptor. This again is a G-alpha S, adenylate cyclase, cyclic AMP, protein kinase A activation sequence. And this is going to activate glycogen phosphorylase through the kinase activity. So the phosphorylase kinase activity will in turn activate the glycogen phosphorylase. Now what if you go into a fight or flight response? Your plasma glucose levels have a possibility of decreasing at a faster rate than normal resting rate. And so the liver has to be triggered not only to maintain normal fasting glucose levels by glucagon signaling, but it also has to ramp up the amount of glucose it's going to put into the blood with epinephrine. And this is going to go through the beta-2 adrenergic receptor, and the scheme is going to be the same as in muscle. We're ultimately going to increase cyclic AMP, activate protein kinase A, and activate glycogen phosphorylase. Now, I'm not making much of a big deal of phosphorylase kinase and glycogen phosphorylase in the liver. And that's because the liver doesn't have any kind of ramp-up mechanism like the skeletal muscle does. Skeletal muscle has a 3-fold increase in ability to control glycogen release. The liver just has glucagon signaling and epinephrine signaling. And these are not different pathways. So this is what we've seen before about synergy and potentiation. Because normal fasting levels gives us glucagon signaling, we're going to activate protein kinase A, and then if we need a little bit more, we're not going to activate a second pathway. All we're going to do is feed into the same pathway and increase the activation of glycogen phosphorylase even more. Okay, so the regulation's a little bit simpler in the liver.
Sorry, question.
Yes?
Is this, I have 2 questions. Is this part of the reason why we're such, as like humans are such good long distance runners? Because of this potentiation and this energy?
This contributes to it. What we also have is fatty acid oxidation, which we're going to look at in a few minutes. And fatty acid oxidation gives us even longer term energy supply. And that gives us the mobility because that's a much greater increase in density of energy. And so that allows us to use energy longer. you're bringing up a really good point about glycogen, because I'm talking about glycogen release in the liver based on 24 hours, it's going to be gone. We're talking about somebody that's pretty much at rest. So if you're exercising, you're going to burn through your glycogen stores in a much narrower window, and then you're going to have to transition into fatty acid usage a little bit more. So that's a really good point you brought up, because this brings us back to our time scale, which is mobile depending on activity and ATP use.
And then, sorry, second question is, I may have been looking ahead too much, but are you also going to get into how the liver in fasting supports itself, or?
Yes,
Okay.
absolutely. That's a huge, huge increase. The reason I say that is because when the liver starts supporting itself in energy needs, at that point, the metabolism in the entire body starts shifting again. Okay, so. Wow, I'm going long. Yes.
Quick question. Is PKA phosphorylating phosphorylase kinase, which then,
Yes.
okay. So
Yes.
it's the same as in skeletal muscle?
Yeah, same as skeletal muscle. It's phosphorylating the kinase, which is going to activate it. Yeah. But it's not such a huge big deal here to worry about in the liver, because all it is just this signal. And typically what most of the books will talk about is that PKA is involved in activating glycogen phosphorylase. So they tend to bypass the kinase in the liver, primarily because we don't have 3 different signaling mechanisms to control it. Now, also in this system here, this kinase is phosphorylating what else? Glycogen synthase. Yeah, same principle here. You need to shut off glycogen synthase, otherwise you're going to reform glycogen. And then you get that futile loop going in there. Okay, so now we're going to talk about fat release. We're shifting a little bit, and the reason I put this in here is because I don't want anybody to have a misnomer that we capitalize on our glycogen stores, and then once our glycogen is gone, then we shift to fatty acid hydrolysis and oxidation. There is no switch like that. Everything is a very, very smooth transition. Glucagon controls glycogen release in the liver to maintain plasma glucose levels, but glucagon is also signaling adipocytes. So the glucagon receptor is also on adipocytes. So we have a simultaneous signal from this hormone to start releasing glucose for oxidation, and then we trigger the adipocytes to start releasing fatty acids. Now the reason that we see glucose oxidation first is because glucose is right there. All you have to do is release it from glycogen stores in the liver, put it out GLUT2, and it's in the circulation. So measuring glucose oxidation can occur right away. With fat release, we have to release the fat from adipose tissue, we have to transport it to the organ, we have to get it into the organ, we have to get it into cells, and then we have to go through a long process of fatty acid oxidation and then generate energy. So we see fat utilization a little bit later simply because it takes longer to get the fat to the organelles for oxidation. So this is what we saw earlier in the fed state. We saw our chylomicrons with ApoB48, dietary fats. Those were coursing through our blood. We were forming remnants because of fatty acid release, because of LPL activity, which is activated by ApoC2. That whole mechanism of binding to HSPGs and localizing this activity. This is releasing fatty acids. In this particular case, the adipocyte is producing the LPL. Because the adipocyte knows that you just ate and it needs some of these fats to store. So then the fatty acids are transported by the fatty acid transport protein across the plasma membrane, and then these fatty acids are assimilated with glycerol 3-phosphate into our triglyceride structure, going through that phosphatidic acid synthesis pathway, and we're going to add 3 fatty acids on the glycerol backbone to make triglycerides. Now, the triglycerides are stored in lipid droplets, and lipid droplets are very, very, very unique organelles. These are actual droplets, and they are coated in a protein called perilipin. Peri means around, lipin means lipid, around the lipid. And so this perilipin coat around this structure is what makes adipocytes very, very unique. So remember I said fatty acids that are stored in any organ, any cell type except adipocytes is pathological. Simply because fatty acids are amphipathic molecules and they can solubilize membranes and damage membranes. So fatty liver disease in the hepatocyte, you build up your fatty acid concentration, your triglyceride concentration, and that's going to solubilize the hepatocyte membrane, causing leakiness and damage. The adipocyte can store an enormous amount of fat because its membranes are protected by this perilipin. So the perilipin coats it. And adipocytes are the only cell type that makes perilipin. So now, we're in a fasting state. So glucagon is going to signal. This is going to go through its normal signaling cascade all the way down to protein kinase A activation. Simply telling the adipocyte it's time to give up the fat because we're in a closed fasting system. So the first enzyme that's going to be activated by pKa is called ATGL. And this stands for adipose triglyceride lipase. And this is going to be activated by phosphorylation, by pKa directly. And this enzyme is going to take triglycerides from the lipid droplet, and it's going to hydrolyze this into a diacylglycerol. And in doing so, we are going to release a fatty acid. The next enzyme is called hormone-sensitive lipase. This also gets phosphorylated by pKa, and this will take diacylglycerol, release another fatty acid, and generate monoglyceride. Now, the monoglyceride structure is still one fatty acid on the glycerol backbone. And we don't want to waste that, so we have an enzyme called monoglyceride lipase. And this enzyme will take monoglyceride to fatty acid and glycerol. Now, these fatty acids are transported into the circulation. Now, because you want the fatty acid quickly for oxidation, for energy, you're not going to waste time building a lipoprotein. So these fatty acids are going to tag along on albumin. Albumin actually has, it can bind about 2 to 3 fatty acids per molecule of albumin. So it serves as a really nice carrier.
And they just like diffuse across the membrane?
Yeah, so it's being transported across by those fatty acid transport proteins, and then it can get through the endothelium by various means, transcytosis or transporters. Now the glycerol, we're going to look at later, but this is going to be under the circulation. The liver is going to take this up, and the liver is going to use this in gluconeogenesis. We'll look at that tomorrow. Now, why is there such a complex cascade? Simply because originally it was discovered that HSL, hormone-sensitive lipase, was the only lipase involved, and all it had to be it was activated. Since then, it was discovered that we have an escalation system like we do with glycogen release. So if you have a little bit of glucagon here, activating pKa, that's going to trigger ATGL to release a fatty acid and diacylglycerol. If that's all the fat you need, you can take the diacylglycerol and turn this back into a triglyceride if you're back into the fed state, and then restore this. You don't have to release all the fatty acids in one shot. Now, what if you need more fatty acids? Epinephrine also has a receptor on adipocytes. Simply because what happens if your skeletal muscle is using so much glycogen that you run out in a short period of time? Your skeletal muscle is going to have to make a hard transition into using fatty acid oxidation to continue energy production. And so here's the source of that fatty acid. This will feed right into the pKa pathway, again, synergy and potentiation, and it's going to increase the amount of flux of triglyceride going through, which increases the amount of fatty acid going out. Okay? Now, fatty acid oxidation. Once we have these fats out on albumin, we're going to use these in metabolically active tissue, except what organ can't use fatty acids? Brain. Brain can only use glucose, and we'll see a little bit later tomorrow or Friday, or tomorrow I think, ketones. So skeletal muscle is a big user of fatty acids. Glycogen is going to be used. The transition between using glucose and fatty acid will be seamless because both of them will come into the cell and both of them will be oxidized. The importance of fatty acid oxidation is that GLUT4 is not on the surface. So even though we have fasting plasma glucose levels being maintained by the liver through glycogenolysis and gluconeogenesis, the skeletal muscle really can't use much of that. So it's going to turn to the fatty acids for energy source. Cardiac muscle. Cardiac muscle has only very, very small glycogen stores. It's designed to oxidize fatty acids primarily for energy. And the way you can tell is if you look at a section of cardiac muscle, you'll see an enormous amount of mitochondria. So any tissue that has a huge amount of mitochondria has a huge capacity for fatty acid oxidation. Skeletal muscle, lots of mitochondria. Cardiac muscle, lots of it. How about the liver? Remember EM pictures, electron micrographs of the liver? If you look in there, it's just packed, absolutely packed with mitochondria. And that's because the liver is going to be using fatty acids for energy for itself during fasting state, because can it use glucose in a fasting state? No, because we're going to see that glycolysis is actually shut off in the liver in a fasting state, because the liver's job is to push its glucose out into the blood from glycogen and make glucose from gluconeogenesis and put that out. So fatty acid oxidation is going to be primary in the liver in a fasting state, and also fatty acids will be used for ketone production. So this is just a reminder of how the fats get into the cell, our fatty acid transport protein. And then our first step is this coenzyme A addition, and that traps the fatty acid inside the cell. Now, we saw that glucose is oxidized by glycolysis in the cytoplasm down to pyruvate, and then pyruvate has to translocate into the mitochondria for further oxidative reactions. Fatty acids are oxidized exclusively in the mitochondria. So that means we have to get the fatty acyl-CoA structure into the mitochondria. So now we have a chemistry problem, again, because we have free fatty acids in the cytoplasm. So what we have is this little structure here. This is a fatty acid binding protein. So all this protein is it actually, the crystal structure of it looks like a clam. Looks like, and it just has a big cleft in it. And the fatty acid, the hydrocarbon tail, just fits in there nicely. And then this protein is a chaperone. And it carries it from the cell surface to the mitochondria. Now, in the mitochondria, we have to control everything that goes in and out of the mitochondria. So the rules here are the same as the rules with pyruvate translocation. Now the way we're going to do that is we are going to take our fatty acyl-CoA. So here is our substrate that we're going to need to transport into the mitochondria. This is the enzyme that is responsible for trapping the fatty acid. So the FATP, fatty acid transport protein, imports this into the cytoplasm, and then we are going to make this coenzyme A addition to make the fatty acyl-CoA. The binding protein, FABP, is what escorts this to the mitochondria. Now the mitochondria has a couple of reactions in order to get this fatty acid inside the matrix. The first one is to take the fatty acyl-CoA and move it across the outer mitochondrial membrane. Now remember, that membrane transport is really not regulated because of these pores. And that's what this is supposed to depict here. These are pores that can actually translocate molecules by simple diffusion. Once it's in the inner membrane space, it's going to undergo a reaction with CPT1, carnitine palmitoyl transferase 1. What this enzyme is going to do is it's going to swap out the coenzyme A for carnitine. And so the product of this reaction is fatty acylcarnitine, and we're releasing a coenzyme A that can now be reintroduced in the cytoplasm for additional rounds of fat delivery inside the cell. Once we have our fatty acylcarnitine, now this is recognized by the carnitine acylcarnitine translocase. So this is the translocase that will take this and bring it inside the matrix of the mitochondria. Now what we're simply going to do is transport this using CPT2, which is carnitine palmitoyl transferase 2. It's the second form of the enzyme. And it's going to reverse the reaction of CPT1. We're going to substitute the acylcarnitine group with coenzyme A, and we're going to make fatty acyl-CoA. Now we've got the substrate in for beta oxidation, and we can generate energy. The reason we do this primarily is because of the coenzyme A. Coenzyme A is either located in the mitochondrial matrix or it's located in the cytoplasm. There's no transporter that will shuttle coenzyme A from either compartment. So we have to go through, which is called the carnitine acyl transporter, to get these into the mitochondria. The other reason for this is this, malonyl-CoA. Where did malonyl-CoA come from?
Synthesis.
Fatty acid synthesis. This is from our citrate that we're going to make acetyl-CoA in the cytoplasm, and then we're going to make malonyl-CoA, And this is going to be used for fatty acid synthesis. So remember I said when you make fatty acids, these fatty acids need to be incorporated into VLDL. We do not want these to go through the carnitine transporter to be reoxidized, because we got a futile loop going on. So we're going to take malonyl-CoA, and malonyl-CoA is going to be an inhibitor of CPT1. So in the fed state, when we have lots of malonyl-CoA, we have lots of fatty acid synthesis, we want to make VLDL to send the fats out to the body. When we're in a fasting state, we want the fatty acids that are coming in from adipose tissue to enter the mitochondria, so our malonyl-CoA levels are very low. What controls the acetyl-CoA to malonyl conversion? Acetyl-CoA carboxylase, ACC. How is this activated?
Citrate.
Citrate activates it? Yep. Well, how about the hormonal activation?
Insulin.
Insulin. So insulin activates this because you want it active in a fed state. What do you think inhibits this enzyme?
Glucagon and epinephrine.
Glucagon. Glucagon primarily. So glucagon inhibits this. And it inhibits it primarily, especially because you want to make sure that if there's any acetyl-CoA stuck in the cytoplasm, you definitely do not want to inhibit the CPT1 in a fasting state. So all fats that come into the liver have to get through this transporter. Now, why do you have to know such an intricate, detailed biochemical transport mechanism? Because there's diseases at CPT1 and CPT2. These are inborn errors of metabolism. So these individuals can rely on only what source for energy? Glucose. Because they can't get the fatty acid into the mitochondria for oxidation. And so these individuals can't use their fatty acids. Carnitine deficiency. Carnitine deficiency is rather uncommon, but a carnitine deficiency can mimic one of these inborn errors. Why is that?
Same pathway.
Same pathway. You need carnitine in order to perform this reaction. So if somebody is a carnitine deficient individual, they may have a fully functional CPT1 and 2 enzymes and translocase, but they don't have the substrate to make the translocation possible. What type of individual might suffer a carnitine deficiency? Where does carnitine come from, our major source? Diet, and what specific type of food?
Protein.
Protein? Sort of. We're getting better.
Animal, cows.
Animal, cows, yeah, meat. Carnitine mostly comes from meat. Now, we can make carnitine, but our pathway is usually inhibited because we get sufficient from dietary sources. So somebody that's an absolute vegan could have potentially a carnitine deficiency. Now that gets into the dietary stuff that I think Dr. Robinson will talk about, being careful with vegans, because some vegetarians and vegans are very careful on their diet, and they're very well balanced on their diet. Others, not so much. They might just decide, well, I've heard that this is a good way to lose weight, I'm going to try it, but they don't have a proper balance in their diet. Okay, so we've got the fatty acid in the cell, in the mitochondria. Now this is where I'm going to boil it down into its bare bones for you, so you don't have to know all the different stages and reactions, because I don't recall any diseases in the actual reactions. So how do we oxidize fat? If we have a fatty acid here, here's our carboxylic acid. Remember, this of course will have a coenzyme A on it. We are going to be performing beta oxidation. So that means this is our alpha carbon here. This is our beta carbon. And so we are going to be chopping off 2 carbons at a time. And these 2 carbons at a time are going to be acetyl-CoA units. So if we have a C16 palmitate, we're going to generate 8 acetyl-CoA. Those acetyl-CoA will go into the TCA cycle just like normal. We'll generate all of our NADH and FADH2. That will go into the electron transport chain, lots of energy production. So what's important of that little picture right there? The concept of energy density. So one molecule of a fatty acid, one molecule of this C16 can generate 8 acetyl-CoAs. One molecule of glucose can generate how many acetyl-CoAs? 2. Do you see why it's hard to lose weight? This is the chemistry right there. It's when you get down to these numbers, fatty acids hold so much more energy. Now, what I didn't show you are the individual reactions. There's 4 reactions that are required to do this cleaving of these 2 carbons off. And not only do you get acetyl-CoAs, but each of these reactions, there's a couple of NADHs that are produced in there too. So there's even more energy than what's being shown here. But the energy density of fat is incredible. Okay? So now what about these buggers? A C19 or a C21. These are also processed by 2 carbons per round of beta oxidation to generate acetyl-CoA. But then in the end, we're left with a 3-carbon molecule. We cannot do a single round of 2 carbons to leave 1 carbon. That doesn't work. So what we're going to do is we're going to turn this into propionyl-CoA, which is a 3-carbon molecule, and then to succinyl-CoA, and then that goes into the TCA cycle. Okay, so that's how we're going to get rid of odd carbons. Now what about unsaturated with double bonds? There's a series of desaturases and saturases that are going to move the double bond if necessary. So this gets into the mechanism of how fats are actually oxidized. But I believe all you really need to know is that if you have a double bond in your fatty acid, we have these desaturases and saturase enzymes that are capable of moving the double bonds into the appropriate position for the final oxidation reaction. It's actually rather complicated on how to do that, but those details I don't think are important. Okay? So we've generated a lot of energy, and this is a little bit of information about to prime you for the case that we have. This case I think we got this one tomorrow or the day after. So carnitine palmitoyl transferase 2 deficiency. So this is a deficiency in the enzyme that converts the fatty acyl carnitine back to fatty acyl CoA. So here's the symptoms, most often in exercise-induced. Recurrent muscle pain, exercise-induced. So how would you interpret that? Muscles use what as their primary energy source? Glucose. Do they run out of their glucose?
Yep.
Eventually. What makes them run out of the glucose faster? Exercise. So then they transition to fatty acids. And with this deficiency, why do they have muscle pain?
They can't do anything.
Because they can't take the fatty acids into the mitochondria for beta oxidation. So they're energy deprived. Weakness, because their ATP levels are decreased, because exercise induces a decrease of glycogen and an increase in fatty acid oxidation. Rhabdomyolysis. That's a weird name. What's that about?
Amino acid metabolism.
Destruction of the cells.
Destruction of skeletal muscle cells. Why are the cells being damaged?
Energy.
No energy. Yeah. Myoglobinuria. That sounds terrible.
Sounds fine.
And that is
Myoglobin into your urine.
myoglobin in the urine. How did I thought myoglobin's supposed to be in muscles. How did that get into the urine?
You just lysed your muscles.
You just lysed your muscles, so it's releasing the myoglobin. Yeah. And all of that because of this deficiency. Now, muscle weakness and pain typically resolves within hours to days, and patients appear clinically normal in intervening periods. Why is that?
Carbs.
Where are the carbs coming from? Diet, fed state, going back into the fed state. So now GLUT4 goes to the cell surface, recharges the muscle, and you have the ability to store more glycogen and fill up your glycogen stores. Wow, how do you fix this problem?
Drink a Coke. Eat sugar.
Eat sugar and never exercise. Oh, I can see this start to grow. Can these people make fatty acids? Oh, yeah. Because that all occurs in the cytoplasm. You don't need this transporter for that activity. What's this compound? Triheptanoid. Why is this an effective therapy for individuals that suffer this disease?
Short chain and medium chain fatty acids can be diffused.
Yeah, short and medium chain fatty acids can diffuse without a transporter. So the CPT, carnitine transport system, is not necessary. And that takes care of the whole problem.